Morning everyone. So today the topic is very much interesting, that is simple harmonic motion oscillations topic, and two special cases of oscillations are already mentioned in the NCERT. We will talk today and tomorrow one by one regarding both of them. So first of all, what we are going to study today is the systems of oscillations, simple harmonic motion, and the NCERT cases. If we want to talk about NCERT cases, here the first case in NCERT is simple pendulum. Simple pendulum, that is the case of angular SHM, angular simple harmonic motion. But you have to understand that this will exhibit angular SHM, the basic conditions of SHM and all those things we will talk about separately. But here this will be showing angular simple harmonic motion only for small disturbances, that is, you can say small angular amplitudes. Small angular amplitude, so that your small angle approximation will be valid, that is theta, sine theta and tan theta are approximately same. Another one is spring pendulum. And the spring pendulum is the case of linear SHM, linear simple harmonic motion, linear SHM. So today we are going to focus on simple pendulum. Our next interaction tomorrow we are going to focus on spring pendulum, all the spring mass systems. Basically here we are going to talk about the spring mass system. Okay. So in this spring mass system, we are basically the common problem for NEET in spring mass system. Common problems are to find out the three things. Number one, in NEET exam, in a spring mass system, they generally ask you X max, maximum extension, maximum compression. The examiner can ask you the maximum during the oscillation. What is the maximum compression or extension produced in the spring? Next question is, what is the maximum velocity of the block during the oscillation? What is the maximum velocity of the block? Everyone knows that the maximum velocity will be attained at the mean position. So you have to identify the mean position, you have to identify the extreme position. The gap between the mean position and extreme position will be the amplitude, and the velocity at the mean position should be treated as the maximum velocity attained. Okay. Number three, time period or frequency. Time period or frequency of oscillation. So this is the way we have to prepare. See, basically when you prepare the oscillations or simple harmonic motion chapter, there are three segments. Number one, general relations. Number two, the graphs based on general relations. Number three, the time period of physical systems. So if you consider the time period of physical systems, those systems can be of various types. But particularly NCERT cases, if we consider, then NCERT mentions two cases. Simple pendulum is an angular SHM case, spring pendulum is a linear SHM case. Okay. Yeah, good morning. Right. So taking you ahead, if we talk about the other things, like for example, let us have before talking about simple pendulum in detail, let us have a brief discussion of linear SHM, then the angular SHM. So we are going to talk about the linear simple harmonic motion. Okay. What is there in linear SHM? We will say condition. First of all, condition. Condition is that restoring force is directly proportional to minus x. Vectorially, if I represent, restoring force is directly proportional to minus x, means linearly related, and negative sign represents that it is restoring. I mean, if you move the body on the right side, the restoring force will definitely try to pull it back towards the left side, or you can say definitely try to pull it back towards the mean position. Similarly, like if you convert the proportionality into an equation, I'm just ignoring writing the vector sign again and again, but you have to remember that yes, we are dealing with vectors here. So it is kr restoring constant into minus x. There are vector signs, but I'm not writing it again and again. You remember that. So this can be MA, that will be kr into minus x. This can be A, will be equal to kr by m into minus x. Here, kr by m, restoring constant must be positive, and mass is by default a positive physical quantity. Restoring constant must be a positive. Restoring constant here must be positive. This kr must be a positive constant. That must be a positive constant. All right. So in order to assure that it is going to be positive and be with dimensional, with no dimensional restrictions, I would say in order to maintain the dimensional consistency and in order to assure that these two are positive, overall constant is positive, we can put it as omega square. We can put it as omega square. By logic of physics also, it will come out to be the square of angular frequency, but we are not directly getting into the logic. So simplicity ke liye we can assume like this. So what we are getting is A will be equal to omega square into minus x or minus of omega square x. This is another condition for simple harmonic motion. Another condition for simple harmonic motion. All right. This was the condition. Right. Now, if you see that carefully, also you can write this acceleration as d2x by dt square. d2x by dt square. This minus omega square x will go on that side and will become plus omega square x. That will be equal to 0. This is called differential equation of linear SHM. This is called differential equation of linear SHM. This side it will be linear SHM. I'll compare it with the angular SHM, so that will become the differential equation of angular SHM. And you can say here time period will be equal to 2 pi by omega, and hence that will be equal to 2 pi under root. Now you see omega is kr by m, so it will be 2 pi under root m by kr, restoring constant. So this is the formula by which you can easily calculate the time period of spring mass system or time period for linear SHM. That is the third aspect. Okay, this we are going to focus tomorrow. Okay, and no NEET paper, I would say in general, that will be framed without a question from the spring. So spring is an integral part of the NEET examination paper. All right, talking about the next one, I am here comparing it with angular SHM. So all the analogous physical quantities will be represented here. How? Now you can tell me this. If I am dealing with the force, translational motion mein, whatever is the role of force in translational motion, who plays the similar role in rotational motion? Whatever is the role played by force in translational motion, which physical quantity plays a similar role in case of rotational motion? Which is the one? The role played by force in translational motion, the similar role is played by which physical quantity in rotational motion? Yes, that's torque. So you will say here, the condition will be, first of all, the condition will be that restoring torque is directly proportional to minus theta, angular disturbance provided. Here x was linear disturbance provided, theta is angular disturbance provided. Okay, angular disturbance provided. If you convert the proportionality into an equation, tau r will be equal to kr into minus theta, and naturally you can conclude that it must be a positive constant. But clearly here the SI unit and dimension, here the units and dimensions of kr and there the units and dimensions of kr are not the same. In linear SHM and in angular SHM, both are acting as a constant of proportionality, both have to be positive, but by units and dimensions, they are not the same or similar type of physical quantity. Both have a tendency, both are called as restoring constants, but the units and dimensions of both the restoring constants will come out to be different. Okay, that is something to understand here. Now we will say the way you can write f as m into a, f as m into a, you can write the tau as I into alpha, and that is kr into minus theta. I into alpha, that is constant of proportionality. Here alpha is angular acceleration. Here alpha is angular acceleration. Okay, alpha. Now you can write there that alpha will be equal to kr by I into minus theta. Kr by I into minus theta. Now here again, moment of inertia. What is I? I is moment of inertia, alpha is angular acceleration. So if you see that carefully, again you can say that just like mass, here is the moment of inertia, just like linear acceleration, here is the angular acceleration. So moment of inertia is a positive physical quantity, and in order to assure that kr is also positive, you can safely represent it that kr by I is equal to omega square. Although this is not the same, this is not the actual logic. Physics logic is somewhat different, but that will involve a bit of complexity. So if we want to make the thing simpler, right, then we can stick with this thing that, okay, we can substitute it safely as omega square. So what we are going to get it as alpha will be equal to minus of, or omega square into minus theta. Omega square into minus theta. This implies, I'm just shifting it a bit here, right? So this implies alpha can be written as double derivative of angular displacement d2 theta by dt square minus omega square theta on this side will become plus omega square theta and is equal to 0. d2 theta by dt square plus omega square theta, that is equal to 0, and yes, that is differential equation. Moreover, if you are interested in time period, that time period is 2 pi by omega, so that will be 2 pi under root I, moment of inertia, by kr, I by kr. I hope the basic thing is clear. What I have tried to, like what is being done here, what I have tried to do is, I would say that I have indirectly told you the procedure to find the time period. How? Sir, linear SHM or spring pendulum, if we talk about, we will displace the particle. We will provide the particle x disturbance from its mean position. We will displace the particle linearly. We will calculate the restoring force. By comparison of the restoring force, we will get the value of kr, restoring constant, and finally in time period we will keep here 2 pi under root m by kr, Now similarly, if we talk about the simple pendulum, which is an example of angular SHM, simple pendulum or angular SHM ke liye, we will still provide the disturbance from the mean position. But linear SHM mein, the disturbance we provided was a linear disturbance x. Angular SHM mein, the disturbance we will provide is going to be angular disturbance theta. So once disturbed from its mean position by theta angle, we will try to write down the expression of restoring torque. Once we write the expression of restoring torque, that can be written as I into alpha, moment of inertia into angular acceleration alpha. Okay. From there, we can get the value of kr easily, and hence the final time period will be 2 pi under root I by kr. I hope these things are clear to you. All right. You can take a screenshot here. I have told you a lot, but the story was completed by the introduction of linear SHM and angular SHM. Let's talk particularly about simple pendulum. So we have understood what is linear SHM, what is angular SHM. Now we are going to talk about simple pendulum particularly. So simple pendulum, and that exhibits angular SHM, simple harmonic motion. Condition of simple harmonic motion, I have already told you that restoring force or restoring torque is linearly related or directly proportional to the linear dependency. It has to be there, linearly dependent to the disturbance provided from the mean position. So exhibits angular SHM for small angular disturbances. Okay. So this property you have to remember. I mean, if you provide big disturbance to the simple pendulum, it will oscillate definitely, but that oscillation will not be a simple harmonic motion. So there is a mathematical condition of linear dependency of restoring force or restoring torque on the disturbance provided, then only it will exhibit simple harmonic motion. All right. So let us consider a simple pendulum. A simple pendulum consists of bob and an ideal string, bob plus string, bob plus string. What is the, if for particularly for ideal string, suppose we talk about ideal string. Here I am just writing string, but there is a term in physics called ideal string. Ideal string has two properties. One is it is massless. So you don't have to consider its motion, string's motion particularly. You don't have to consider string's momentum, string's energy. You don't have to consider the energy stored, because kinetic energy of the string, I want to say. Secondly, it is inextensible. So because of the load, there will be no extension provided in case of simple pendulum. There is no extension produced in that. So bob plus string, and bob is generally, practically, we say bob, you can call it a heavy metallic ball like that. Okay. So you have seen that n number of times in the labs. So I'm going to talk about this arrangement first. So this is P point. P point is basically point of suspension, from where you have suspended it, hanging it. Okay, point of suspension. L length of string, M mass bob. It's a very light string. Ideally it is massless, practically it is a light string, and practically it is a bit extensible also. So what will happen to the extension of this, we will talk about it one by one. But here this is the mean position, MP. You can call it mean position. What is the feature of the mean position? That without any disturbance, if you leave the system, where the system will reside. So you can also call it the most stable position. You can also call it the equilibrium position. So if it is equilibrium position, you can say the net force on it is zero. And since it is most stable, the potential energy has to be minimum. I am denoting the potential energy by U. So its potential energy has to be minimum. So mean position, without any disturbance, you leave the system. Wherever the system resides, you can commonly, these things are generally common. See, basically there are a lot of terms you need to understand before understanding of this topic or this chapter. But I am trying to keep it a bit simple for you. So that's why I would say that you can take the things in general so that you can relate with it from day-to-day life, right? So that's our motto. Then we can get into the in-depth concepts also one by one. So here, this is the mean position. The first step I have told you is disturb the system from the mean position. Since it is a system which will exhibit angular SHM, you should not try to disturb it linearly. Moreover, even if you try to disturb it linearly, it will automatically rotate. Even if you try to hold the bob and try to move it forward, it will automatically try to move it in an angular sense of anti-clockwise sense. Okay. So let us assume that you have provided it some disturbance theta. Okay. And after providing this disturbance, angular disturbance theta, you can see that this is the orientation. Here is the M mass bob. Now we will try to mention the free body diagram, the forces acting on it. So you will say here is the force acting, W equals to mg, and here is the tension acting, tension T. Okay. I will say that you can resolve this mg in two senses. So one will be like this angle is theta, so this angle will be theta again. So you will say this component will be mg cos theta and this component will be mg sin theta. So at this position, like in this situation, your T will be balancing the mg cos theta. You are just providing it a disturbance slowly and you are releasing it. You are releasing it from here. So if you are releasing it from here, there is no velocity. U is 0. Okay, you just dropped it. So U is 0, so there is no centrifugal force acting that you have to consider. So clearly you can say T will be equal to mg cos theta, and this mg sin theta is going to provide the necessary restoring torque. This mg sin theta will provide a restoring torque. So I will say here the restoring torque, tau restoring. I'm just writing the magnitude because you can clearly see that you have provided an anti-clockwise disturbance and the torque is trying to make it clockwise, bringing it back. Okay. So naturally you can say that it is a restoring one. So I'll ignore the plus minus sign and I'll simply write the magnitude. So that is going to be equal to what? That is going to be equal to the component of force which will be resulting into a torque. Restoring torque is mg sin theta. And if you extend its line of action of force, the perpendicular distance from point of rotation or point of suspension is L. So if you write it as F into D perpendicular, so that is going to be equal to mg sin theta into L, mg sin theta into L. If you see that carefully, it is mgl together into sin theta. So you can say here clearly, your restoring torque is directly proportional to sin theta, but restoring force is not directly proportional to theta. It is not directly proportional to theta, so not an SHM, because the condition for simple harmonic motion is that the restoring torque is directly proportional to the angular disturbance provided. So you will say not an SHM in general. So the motion of simple pendulum in general is oscillatory. The motion of simple pendulum in general is oscillatory motion, but it is not a simple harmonic motion. I hope the fact is clear to you. But sir, here you have assumed that you are talking about small angular disturbances. That's which plays the key role. Because if you talk about general disturbances, it will be a general oscillatory motion, but it will not be a simple harmonic motion. And if you want to talk about the simple harmonic motion, definitely the angular amplitude should be very, very small. Angular amplitude should be very, very small. So now I'm going to start with it. I will say for small angular disturbances, for small angles, sin theta is approximately equal to tan theta is approximately equal to theta, small angle approximation. So can I say the restoring torque, which is mgl into sin theta, can be approximated as mgl into theta. The moment you will write mgl into theta by comparing with tau r is equal to kr into theta, we get by comparison whatever will serve as a coefficient of theta can be taken as a restoring constant. That can be taken as a restoring constant kr. So you will say kr value is equal to mgl. Sir, what we are exactly trying to do? We are exactly trying to find out the time period of simple pendulum. So if you notice, simple pendulum is a type of angular SHM, and in angular SHM, the time period is given by this formula. I have related all the aspects, all the basic relations that could be used in the exam. Okay. So if you are talking about the time period, we need two things, moment of inertia as well as kr. The value of kr we have calculated it here, and the moment of inertia calculation is also very easy. Also, the moment of inertia of the bob about P point is md perpendicular ka square, md perpendicular square. So in better way, we can write it as m. What was the perpendicular distance of this from there? The bob mass m from the point of suspension, that is L. We have used it earlier here also. See, d perpendicular. So I will say this is ml square. So finally, the time period will be 2 pi under root I by kr. So I is going to be ml square by kr is going to be mgl. If you look at it carefully, you got the final answer as 2 pi. See, ml from the numerator and single ml from the denominator will be cancelled. It will be L by G. It will be L by G. So moment of inertia is this, restoring constant is that, and finally the time period of simple pendulum is 2 pi under root L by G. 2 pi under root L by G. But dear, if you remember, I have already mentioned it several times that this will exhibit simple harmonic motion only for small angular disturbances, which simply means that here this time period 2 pi under root L by G will be applicable only for small angular disturbances. Alright. Today I am not taking you to too many NEET PYQs or problems like that. Today we are trying to understand the different aspect of this oscillation chapter, very much important for the NEET exam. And oscillations may be particularly the NCERT 2 cases. One is simple pendulum, one is spring pendulum. We have taken the first case today, that is simple pendulum. Another thing we will take it up in the next interaction, that will be the spring pendulum. So this is the first highlight. What is the simple pendulum? How we are sure that it will exhibit the simple harmonic motion, and assuming or taking the approximation that it is valid for simple harmonic motion, then how you can find its time period. This was the target which we have achieved on this slide. Okay, so I hope you have a clear-cut idea of simple pendulum now. Now there will be variation in the time period. Variation in the time period of simple pendulum. Now we can clearly see that time period is 2 pi under root L by G. Time period is 2 pi under root L by G, and that is interestingly coming out to be independent of mass of bob. Independent of mass of bob. That doesn't depend on mass m. Okay, so if you want to vary the time period, you can either talk about the variation in length or you can talk about the variation in g, acceleration due to gravity. So case 1 I am talking about. Case 1 is variation in the value of variation due to length. Variation due to length. And then I think whenever next time we will be meeting, we will be talking about case 2, that is variation due to the value of g. Due to value of acceleration due to gravity. As you already know that acceleration due to gravity value also is not same everywhere. It varies for different planets. It varies on the surface of Earth with rotation, different on equator, different on poles. It decreases with elevation as well as it decreases with the depth. So the value of acceleration due to gravity will definitely keep on changing. Since it is coming in the expression of time period, definitely it's going to affect the time period expression also. Okay, but that is not our concern right now. Let us start with case 1 first. Moreover, it reminds me of another problem model also that deals with the errors. So here if I write t is equal to 2 pi under root L by G, sometimes this also is used in calculation. This expression is also used in calculation of G, and I would say rather error calculation in the value of acceleration due to gravity. So whenever you are dealing with, because the first chapter that is units and measurements, that also has a very important section which gives you one problem in every year exam. So blindly you can say that error propagation and combination is a topic from which the final exam will definitely bring a problem. Okay, so error calculation of G, how to use this expression in order to study this topic or in order to calculate the error in the value of G. So here we write it as, like if you square both sides, you can write t square is equal to 4 pi square L by G, or in a better way you can write G will be equal to 4 pi square L by t square. So if I apply the power rule, so I will say percentage error in the value of G, percentage error in the value of acceleration due to gravity. 4 pi square is a mathematical number, mathematical constant, so it will not involve any kind of error. The coefficient of L is 1. Here you will say G is directly, the power of L is 1, sorry, not the coefficient. Coefficient is 4 pi square, that will be a number. It will not involve any error. The power of L is 1 and T is having 2. So what you can say is percentage error in the value of G, that will be 1 time percentage error in L plus 2 times percentage error in T. So shouldn't you write it minus here? No. If you are dealing with percentage changes, then you should consider plus or minus depending upon the sign of the power. But errors never compensate each other. That's why NCERT also gives you the heading of error propagation. Errors always propagate. Errors are always added. they never compensate. Errors are never subtracted. So I should say that that's why I'm saying here that errors are always added, and hence I should say that percentage error in G will be 1 time percentage error in L plus 2 times percentage error in T, because in case of percentage error or fractional and relative error, you simply multiply the power as a coefficient of the error. Okay. Now, talking about the variation due to length, variation due to length will also come because of the two reasons. Number one, it might come due to thermal expansion, extension due to thermal expansion. Okay, you rise the temperature, the length will definitely change, it will be extended, hence it will affect the time period. So this is common for the NEET. Number two, extension due to load. Because M mass is not coming in the formula, but you cannot ignore its presence here. In fact, one of the component of weight was balanced by the tension, and another component of weight acted as a that provided the restoring torque. So definitely gravity is involved here. You cannot ignore the gravity. So I would say that because of the load, and it's not an ideal string. If it would be an ideal string, then you will say it is inextensible. But in case of real string applications, we are talking about practical physics. So we will say due to load, there will be some extension produced. And if there will be some extension produced here, that is extension due to load. You can consider its Young's modulus, this, that, many things. But here I will say this is very rare for NEET. And if it is very rare for the NEET exam, we are not going to discuss it right now. At the end, just before the exam, we can, for a safe side, we can have a look. But as of now, we are not considering it. Okay. As of now, our concern is only to talk about thermal expansion. So suppose I have raised the temperature theta. Then can I say that here final time period will be equal to 2 pi under root L final by G, which can further be written as 2 pi under root thermal expansion. I hope you remember L final is equal to L initial. L final is equal to L initial 1 plus alpha delta theta, like that. L final is equal to L initial 1 plus alpha delta theta. That's what the basic formula of thermal expansion I'm going to use here. I can simply write it as L initial 1 plus alpha delta theta by G. So here it is 2 pi under root Li by G, and it is 1 plus alpha delta theta taken separately. Root means its power half. And as far as you remember, I hope you remember it, that thermal expansion, in all the cases of thermal expansion, you always have a habit to apply the binomial approximation. So this power will get easily multiplied here because practically the value of alpha is very, very small in comparison to 1. So you can write it as T final will be equal to T initial. This 2 pi under root Li by G can be written as T initial, and it is 1 plus half alpha delta theta. So this is the governing relation you are going to use here. This is the relation. Also, from the same relation, if I try to calculate here delta t, which is T final minus T initial, then I just need to open the bracket here. So this will be T initial, sent on that side it is T final minus T initial. Also it will be multiplied with the second term. So that will be T initial half alpha delta theta. And common question in the NEET exam is fractional change in time period, delta T by T initial. Fractional change in time period, fractional change in time period of simple pendulum, that will come out to be half alpha delta theta. Half alpha delta theta, this is very much important here. This step particularly, this expression is very important. So I'm telling you what is not so important for the NEET exam as well as what is quite important for the exam. So this is something very important. You can remember it right now as a new formula. And once you get the expression of fractional change, it's very, very easy for you to talk about the total change. Chalo, we'll restrict ourselves here up till fractional change only. When from the fractional change, how to calculate the total change in a given duration, say it a day, say it a week, say it one complete year, that we will deal with some interesting questions based on simple pendulum very soon. I hope the story of simple pendulum is partially completed and understood to you, because the other case, that is variation due to value of G, that will involve a lot number of cases. Okay? There will be many small cases talking about pseudo force, keeping the pendulum inside a swimming pool, keeping the pendulum in a cart, then accelerating the cart, say it horizontally, vertically up, vertically down, on an incline. Many cases will come. Okay, whenever you can vary the value of acceleration due to gravity, but that we will take in the, once you come to the campus, then in the live lectures we will be talking about it. As of now, you can stick to the variation caused due to the value of L, length. I hope the simple pendulum is clear, and SHM is an integral part of your final NEET exam. Majority of chances are there that the question will definitely come from the spring mass system, and a good number of problems also come from the simple pendulum. So in our next interaction, our target topic will be that most predictable problem model for the NEET exam, that is going to be spring mass system. All right. So that's all for today. All the best. Bye bye.
Good morning, one and all. In our today's botany session, we are going to discuss our next vegetative organ in angiosperm, that will be the leaf, right? I hope so you have revised the previous topics regarding the root and the stem. There we discussed their general features, and even we compare somewhere where the differences are lying between root and the stem. Now next important organ, we are going to take the leaf, right? Let's talk about generally about the leaf here. Can I say this leaf is an important vegetative structure of the flowering plant, being it is helping in the photosynthesis. Can I say this is the manufacturing unit for the flowering plants, where it is manufacturing the food for all the organs, non-green parts I must say, for them it is forming the food. So what are its general characters? For that thing, usually my habit for this introduction of the general feature, I am giving one mnemonic for that, right? What is that? That is the God's leaf, right? Where G stands for, it is green, obviously photosynthetic, vegetative part of the flowering plant, right? And it is green, obviously they will perform the photosynthesis, right? Photosynthesis word, sometimes they are writing the assimilation, right? And now O stands for that this organ originates, it is taking about the concept of its origin. It originates from shoot apical meristem. Right? D stands for this organ is dissimilar or that differs from species to species. It will differ. Means what is its texture, what is its type of lamina, what is the shape of that, what is the margin, what type of margin it is having, how much incision it is having, that all varies from species to species. So it is dissimilar. Then even as here I mean to say, they carry the small opening which you call it as a stomata for the exchange of gases, right? Then even L stands for it is having limited growth, or even you can say from L it is the lateral expansion of the region where different structures you will find, like roots, stem, they are elongated and cylindrical structure, but leaf is going to be the expanded part, being it is like a compressed thing. So it is going to be lateral expansion part, and here it is having the limited growth. It not keep on growing throughout life. It cannot do like that. Overall plant can show unlimited growth, but not the leaf. Now here E stands for that it is exogenous in its origin, right? And here A stands for their acropetal arrangement. Acropetal, earlier even I said you younger are towards the tip of the stem, whereas older are towards the base. And what is present at the axil of the leaf? It is carrying axillary bud. Right? In my yesterday lecture, I said you, like suppose if I am taking here the leaf, suppose this is the stem part and this is the leaf part. Right? On its axil, it is having a bud. Right? This bud, you call it as axillary bud, which is a leftover of the shoot apical meristem. Right? So this axillary bud is, even the presence of this bud is telling you that this is the leaf as an organ. Now coming to the next part here, F stands for what thing? Here F stands for, generally this structure is flattened. Right? Why generally word is written over here? See, in your NCERT book, these words are very important. Generally, most, almost, few. So be careful about that. Generally, it is flattened. Means some leaves are there which are not flattened. For example, like we have spines on the cactus. It is not a flattened structure. It is going to be elongated and pointed structure. It is the leaf modification spine. So in that case, it cannot be a flattened structure, right? So it depends upon the type what we are discussing. But generally, if you see, we are discussing journal characters. So it will be flattened. So these are some of the journal character you must know about the leaf. Now let's talk about the parts of the leaf. How many parts basically we are studying in a leaf? 3 parts. First is the lamina. Second, we will discuss about the petiole. Third, we will discuss about the leaf base. Now first I am taking for the lamina. Can you see this is the most conspicuous? Conspicuous means clear. This is the conspicuous. Conspicuous means it is clear and, or you can say prominent part of the leaf. So this lamina part is flattened part, and even it is main photosynthetic part over there, and it is supported by veins and the veinlets. Veins and veinlets will be there. Like suppose this is the midrib and here are the veins and further veinlets are there. So this lamina is going to be supported by all these veins and veinlets. These veins and veinlets will be acting as a channel for the conduction of water, minerals and the food. Being it is the manufacturing unit of the food, from the leaves to the other non-green parts, that channels will help to going to give that. But from the roots, it is picking up the water and minerals for this organ, right? Now, what is this part? This part of the lamina, this part, this one, this all margin, this all you call it as what? Leaf margin. What is at the tip? Obviously, tip of the leaf, or you call it as a leaf apex, right? On the leaf margin, can you see these cuts are there? It cannot be always. Sometimes it is continuous type of thing. It is not having this, you can say this is not having these cuts. These cuts you call it as incisions, right? Incisions depth will be how much? That will say you the category of the types of the leaves as well. If these incisions are not there, that means the lamina is entire. The lamina is entire. Entire lamina will be there. But if incisions are there, that will decide the type of leaf, whether it is a simple or compound. That we will discuss after this topic. Now, coming to the next part, what is the petiole? Can I say this is the long flexible structure which you will learn in the anatomy part. The petioles are carrying collenchyma, which is giving the flexibility, and why this flexibility is required to expose this leaf towards the light, and even very important thing regarding flexibility, that it flutters on the air, means it will brings the cool air on the, or it will maintain a cool environment on the surface of the leaf, and add on to that it brings, I must say here, it will keep the surface cool, and along with that it brings fresh air on the surface of the leaf, right? And exposure even will be there. Sometimes petiole can be absent in the leaf. Such type of leaves you call it as the sessile leaves, where petiole is absent, right? For example, in case of calotropis, right, you will find sessile type of leaf. Petiolate leaf will be much common in our environment. For example, like hibiscus, it is petiolate leaf, means it is having the petiole over there. Now next part is the leaf base. Leaf base is what? This is the point at which the leaf is going to attach on the main stem. So this is a point of the connection of the leaf with the main stem, right? And now what is there? In the leaf base, you will find some modification, and that is very important question on the NEET, that you call it as pulvinous. Next you call it as a leaf sheath, right? Pulvinous is in case of the legumes, whereas leaf sheath is in case of monocots, right? Now what type of thing the leaf base is? This is like a point of connection of the leaf with the main stem. Now what is this pulvinous? Pulvinous, when you see the leaf of the legumes, you will find at the base some swollen cushion like structure is there, that you call it as pulvinous. It is a feature of legumes. Whereas in monocots, you will find when the leaf is coming out, but its base is covering the stem completely or partially, that structure you call it as a leaf sheath. So that is a modification of which part of the leaf? That is of the leaf base, right? This is very important question based on your NEET exam, that what is pulvinous and what is a leaf sheath. And do remember it example, legumes are carrying the pulvinous, whereas the monocots, they are carrying the leaf sheath around the stem, which is the modification of the leaf base, right? So I hope so you had got the little bit idea for the parts of the leaf. And now we are going to take the next topic. What is that? The venation. Just now I said you the lamina is supported by the midrib, veins and the veinlets. So what is the venation? The arrangement of veins and veinlets in the lamina of the leaf, that you term as a venation. Now this word is venation. This word is not the vernation. This is not the vernation. What do you mean by the vernation? Arrangement of the young leaves, that you call it as a vernation. But here we are talking about the venation. So don't get confused between these terminology, right? Now let's talk about the types of the venations. So two types of venations we are having in our plants: angiosperms, reticulate and parallel venation. What is reticulate venation? In the reticulate venation, the lamina is having in the middle the main vein. The main vein, we call it as midrib, right? Now further this midrib is divided into the veins, right? But the main vein you call it as midrib. Midrib further into the veins further. Now veins are further divided into veinlets, right? Now being it is reticulate venation, the further reticulation you will find here, the further they keep on dividing and redividing. That division and redivision, that you call it as the reticulation. It is a sort of network formed by the minute veins, right? So what we are discussing about reticulate venation, when veinlets are very clear and further it is dividing into the network of this reticulation, such type of venation you call it as reticulate venation. Here it is generally present in case of dicots. Exceptions are there, but you must know major this is a feature of the dicots. What is the parallel venation? In the parallel venation, the leaf, no doubt, it is having the midrib as a main vein, and to this main vein other veins are going to be parallel, or you can say, if we are having condition like this, one more, in other cases this is the midrib and the veins are parallel to each other. Even this is which type of venation? Parallel venation, right? So main thing you must remember here, what is a parallel venation? Where the veins are parallel to each other. That type of venation you call it as parallel venation, right? But in reticulate venation, what is happening? The reticulation is there, right? That reticulation is absent in case of parallel venation, right? Even the parallel venation can have veinlets, but it is not conspicuous, it is not clear. But what is left now very much clear in a parallel venation? The midrib part and rest of the veins, right? Even here midrib part and these veins. So what is parallel venation? Where veins are parallel to each other and the reticulation is absent. And generally it is present in which case? Generally it is present in case of monocots. So please do remember that not in all monocots. Again, exceptions are there, right? So two types of venations you will find here, reticulate and the parallel venations, right? So one topic we have discussed under the leaf, that is a venations. Now let's talk about the types of the leaves, right? These types of leaf is based on the incision. Earlier just now in this lecture I said you incision is decided based on the margin, that how much cut is there deep inside, whether it is touching to the midrib or not. Like for example, let me to take the slide. If I am taking the leaf as such, can you see it is showing the entire leaf margin? If there is entire leaf margin, such type you call it as simple leaf, right? But even if the margin is showing incision, here what is there? Incision. But here incision is not touching to the midrib. This is not touching with midrib, right? Can you see this incision and this is the midrib? It is not in that much depth that it will start touching to the midrib. Again, this is which type of leaf? Simple leaf. Even this is which type of leaf? Simple leaf, right? So such type of leaf where incision is absent, or if incision is there but not touching to the midrib, such type of leaves you call it as which type of leaf? Simple leaves, right? Now what is the compound leaves? In case of compound leaves, the incision is such that, like this incision is so much in depth that it start touching to the midrib, right? If it is started touching with the midrib, can you see it give out a minute type of structure which you call it as leaflets, right? This is not a leaf. These are what? Leaflets. And this now complete is one leaf. How we can say this complete is a leaf and these are the leaflets, because leaflets will not have buds at their base. If I take this complete as a leaf on a stem, so on its axil bud is present, but bud will be absent at the base of the leaflets. On that grounds, I can say this is a compound leaf having the leaflets, right? So do remember, this is important here. We are discussing compound leaf. Or even compound leaf can be like that. If this is a petiole part, which is a stalk of the leaf, here the leaflets are coming out with the deep incision and making the point of joining at the common point. What this common point? That is a tip of the petiole. When the leaflets, they will going to touch, due to this incision, they are touching to this common point. This you call it as common point. What is this common point? That is the tip of the petiole. When all these incisions are so much in depth that these leaflets are grown in like a form of palm, like in hand, such type no doubt is a compound leaf. But which category of the compound leaf? Palmately compound leaf. Which type of leaf it will be? Palmately compound leaf. This one you call it as pinnately compound leaf, right? So how many types of the compound leaves are there? Two types, pinnately compound and the palmately compound, right? Now let's talk about the example here for simple leaf, like entire what we have shown. This example can go with the mango, right? And such type small incisions when you will find like this, chrysanthemum. This example is not given in your NCERT. Just to complete all the examples I have written there. If you are feeling burden, you can leave this example here, right? No need to learn, but just to mention you the example I have written. But now important example here for pinnately compound, you must remember here it will be including rose, neem will be the example here. Here will be the silk, cotton will be the example. So do remember all these examples comes under which type and which category of leaves. Let me to show you through some pictures here, like this one shown in your book. Can you see here types of leaf again? This is a simple leaf. Can I say it is having entire leaf margin, right? And this is which type of leaf? Written over there, pinnately compound leaf. This is what type? Palmately compound leaf. Can I say both comes under which type of leaf? Compound leaf, right? And in this compound leaf, the midrib now we call it as a raches. Here this midrib now in pinnately compound you call it as raches. Raches is a foreign language term where that we means spine, like spine is there, and in the same way here this is sort of a spine for a compound, pinnately compound leaf, and supporting these leaflets. Even this will be the leaflets, and even from this side these are the leaflets. And you will find axillary bud here, not in the leaflets. Here even the axillary bud will be there. Here even axillary bud will be there. And do remember these axillary bud, one more point I am adding here, these axillary bud will be guarded by a tongue-like projection which you call it as stipules, right? Let me to show you this point here itself. I have drawn this diagram earlier. There this axillary bud is covered up by this stipules. These are scale leaf-like structure which is protecting the axillary bud, right? So a pair of stipules can be there at the base of the leaf, and usually students get confused between the stipules and the bracts. Bracts you will find at the base of the stalk of the flower, but stipules you will find at the base of the stalk of the leaf. What is the stalk of the leaf? You call it as petiole, right? And stalk of a flower you call it as pedicel. What is at the base of pedicel? Bract will be there. What will be at the base of the petiole? The stipule will be there. But what is the function of this stipule? It will protect the axillary bud, right? Even leaf base, in the case of the parts of leaf, it even helps the protection of the leaf base, right? Okay, so now coming to the next topic of discussion for the leaf, that is the phyllotaxy. See here, the word phyllo stands for the leaves. Taxis word means arrangement, right? So here, that's why definition comes out to be, it is a pattern of arrangement of leaves on the stem or branch. How the leaves are going to be arranged. Why different species show different arrangement? Reason, they need the proper exposure towards the light. Every leaf from tip to base may not get proper exposure. To get a proper exposure, they must have a proper orientation or arrangement on the main stem or on the branch, right? Now let's talk about the types of phyllotaxy that we have to discuss now. Okay, now what are the types of the phyllotaxy? Basically, three types are there. One is the alternate type, second is the opposite type, third is a world type, right? Now let's talk on first alternate type of phyllotaxy. In the alternate type of phyllotaxy, you will find this is a stem now. Just now we have completed the types of leaf. So my request, please don't consider that as the midrib and the leaflets. No, this is the stem or a branch on which what have been arranged, leaves are arranged. Now we are discussing the leaves arrangement, not the leaflets, right? So be careful about that thing. Alternate type of phyllotaxy, you know that the leaves develop at the node. It originates from the shoot apical meristem, but it develops on the node part. So can I say here, this is a node part carrying one leaf, second node carrying second leaf, and so on, alternative to each other. If I tie a thread here, you will find if I make it go like this, it will show you like a spiral winding around the stem. So that's why sometimes we can call alternate type of phyllotaxy as spiral phyllotaxy, where leaves are arranged in alternate manner, right? Now do remember, in alternate type of phyllotaxy, each node will carry, right? So here each node will carry one leaf. Here example will be of China rose, mustard, and sunflower. These all examples are from your NCERT book. Do remember, all the three examples written now in front of you on the board, they are all three from different families. Now you must know China rose is in which family? Malvaceae family. It is newly added family in your syllabus. Mustard belongs to Brassicaceae family. Even it is newly added completely, you have to learn now. Sunflower belongs to Asteraceae family, which you even call it as Compositae family, right? So you must know based on families even that which type of phyllotaxy is there. So be careful, you need not to go beyond to all these families, but what is newly added, what is previous, based on that you should know within chapter which examples have been given. Now coming to the next one, opposite type of phyllotaxy. You will find here, can you see here node, but how many leaves are there? 2 leaves on each nodes are there. Even here one node, how many leaves are there? 2 leaves are there. So in opposite type, each node will carry how many leaves? 2 leaves are there at each node, right? But why 2 diagrams have been shown here? When you will see in the first diagram, suppose I am taking here for guava. In guava, you will find, like if I am taking this as a one pair, second pair is going to be parallel to the next, parallel like this. But in this case, one pair will be like that, second will be perpendicular, right? Next will be perpendicular, right? So this perpendicular arrangement, one pair to the next, that will show the one type of the opposite type, and this is the another type. Here the example will be the calotropis, right? When one pair is going to be parallel to next, such type you call it as superimposed type of phyllotaxy. If it is perpendicular to each other like this, such type in calotropis you call it as decussate type of phyllotaxy, right? So do remember at least the example that Gava and calotropis will show which type? Opposite type. And opposite type, each node will have how many leaves? 2 leaves are there, right? So this is important. The examples and each node, how many leaves will be there in which type of phyllotaxy. Now coming to the world type. World type means in each node, can you see more than 2 leaves are there? In world type, each node will carry more than 2 leaves, right? Here example will be Allistonia, Nerium, very important examples. But here hidden thing is what? Nerium, you should know, maximum it can have how many leaves at one node? 3 will be there. Allistonia will have more than 3. Allistonia will have more than 3. If they will ask you in world type more than 3 leaves, then you should not give the example of Nerium. Then you have to give the example of Allistonia. If they are simply saying more than 2, then you can quote both the example Allistonia as well as Nerium, right? So this is next important thing you must remember, and they can convert this information into numerical part as well. Like I am giving you a small example, like a person who is collecting the different branches of different species, taking off the twig of one twig of a Gava which is having 5 nodes, and second twig it is taking off from the calotropis which is having 6 nodes. And next one example, it is keeping from China rose, one twig, twig means branch, right? One twig from there carrying 5 nodes over there. Allistonia, one twig or a branch which is carrying the 2 nodes. Now calculate the total number of leaves over there. If you know in which type of phyllotaxy, how many leaves are there at each node, if you know this information, then you calculate the information, multiply with number of nodes, and then take a total of that, right? Let me to give you one example. I am writing here on the board for you. For example, a twig of Gava which is containing 5 nodes, right? A twig of China rose which is containing, okay, I am taking 5 nodes again here. A twig of Allistonia with 2 nodes, but here suppose I am giving each node is carrying 4 leaves. Suppose I am taking that, right? Now you have to calculate the total number of leaves. Here you know that Gava is having at one node 2 leaves. So 5 into 2, 10 leaves here. Right? China rose is having 5 nodes on a one branch. Twig, I want to say, that is a branch. Right? So one branch, 5 nodes, but one node is having 1 leaf. In the China rose being alternate type of phyllotaxy, 5 leaves will be there. Right? Allistonia, 2 nodes, but it will be having 4 leaves, 8 leaves over there. Righty? So here we have to take it, 23 leaves will be there total, right? So this will be your answer, right? Total leaves comes out to be 23. Just check it out once. I've just given you a rough idea that this concept can be correlated with the numerical part, right? Now coming to the next part, the leaf modifications. This is our last topic to discuss on the leaf, and although this topic is not there in your new book, but you should go through that points as well of leaf modification. The first modification is not helping in primary function of leaf. What is the primary function of leaf? Photosynthesis. Second important, exchange of gases through the stomata. These two are the main functions of the leaf, right? Very important. If the leaf structure modified and perform the function other than primary, that you call it as leaf modifications. Like here, the first function, tendrils are giving for the support. Now here example will be of P. Please do remember stem tendril. We discussed there, for example, grapevine. Grapevine tendril is a stem modification. In the P, tendril is the leaf modification, right? It's terminal leaflets. Terminal means which is at the tip. At the tip, the leaves, or you can say they are leaflet because it is having pinnately compound leaf. So there the leaflets, they will modified, they will you can say modified into this structure which you call it as tendril. Long cylindrical part which is coiled like a spring help the pea plant which is weak to get a support, right? So this is one function other than the photosynthesis or the exchange of gases. Here we take it as a support. Do learn the example P. Next modification, we call it as phyllodes. And don't confuse it with phylloclade and cladode, which we had discussed in the stem modifications. Now phyllodes is the leaf modification. Here it helps in the photosynthesis. Now you are surprised that we are discussing the leaf modification. It is primary role is photosynthesis, then how come this is a secondary function? Because this photosynthesis right now in phyllode is not performed by lamina. It is performed by petiole now. That's why it is the modification, right? So in phyllodes, the petiole become expanded and leaf-like, and the leaf-like. That's why we call it as leaf modification, right? And this type of structure you call it as a phyllodes. What is an example over there? Australian acacia. This will be the example for this, right? Next, spines. Spines, don't confuse it with thorns. Both are for protection. Which function? Now it is performing protection. Example here for cactus. Under that, opuntia or euphorbia will be there, where leaves are modified into spines for the protection of the phylloclade, which is the stem modification and which is preserving the food over there. And spine even help to reduce the transpiration by reducing the surface area of leaf, right? So next modification we learn based on the protection, right? Now let's check on the rest of the modification. Even this bulb is for storage. We discussed bulb in the stem modification based on discoid, compressed type of the stem. But who is storing the food over there? Leaves are storing the food. So here the leaves will store the leaf, and what function it is performing? Storage here in case of like onion and even in case of the garlic, right? Two examples will be there. Even it can perform the function for capturing of insects, right? So here, for example, pitcher plant, right? So which example we are quoting here? Pitcher plant. So this pitcher plant, its lamina is modified into capturing organ, right? Whereas the leaf tip or the apex will converting into the lid. This lid is going to close. This lid is the modification of leaf apex. Lamina is modified into this capturing organ, that is in pitcher plant, right? So why these insectivorous plants are capturing the insects? Because they are surviving in that type of soil where there is deficiency of nitrogen, and that nitrogen deficiency will be overcome by taking or capturing these insects. That's why they are capturing the insect. So this was the last modification in context to the leaf we have discussed. In this way, we have completed our next vegetative organ, that we call it as a leaf, right? Just go through that topic gradually, learn the examples, that's very important. I know that if you are repeaters, it's very tough to listen again and again all these concepts, reading that, but definitely if you take it for granted, then you will make the mistakes. Make a habit to listen, to revise, to recapitulate the things. It is more important, right? So we have completed with the leaf the vegetative morphology from the morphology of flowering plants. That's all for today. Take care. Thanks a lot, students.
Good morning to all. Welcome back. So today our topic is generation and conduction of nerve impulse. First thing is here. In the previous class also we have discussed here, neural tissue contains two types of cells called neurons as well as neuroglial cells. But the neurons are excitable cells here. So the same point, neuron is an excitable cell. What is the reason? This is actually assertion and reason question here. So neuron is an excitable cell because their membrane is polarized. This is very important point here. So why do the neurons are said to be a excitable cell? Means reason is their membrane is, their plasma membrane is here in a polarized state. What is the meaning of polarized state? Means here, polarized state means when the neuron is in resting state. Imagine when the neuron is not under stimulus, it is present in the form of resting state. Whenever the neuron is present in resting state, that time, suppose this is axon, and this is known as axolemma because this is membrane. Plasma membrane of axon is known as axolemma. So whenever the neuron is in resting state or the membrane of the neuron is in resting state, that time this membrane will be, this point also very, very important point. So during this resting state, the membrane is permeable. The membrane will be permeable to potassium ions only here. Okay, suppose this is the plasma membrane or axolemma. When the membrane is in the resting state or without any stimulus, that time through this membrane the potassium can diffuse here. The membrane is permeable to potassium ions like this here. Whereas during the same resting stage, the membrane is impermeable. This is actually permeable and this is impermeable. It is impermeable to, number one, sodium ions. It means the sodium ions, they never permits when the membrane is in resting state here. So resting state means that is known as polarized state here. So it is nearly impermeable to sodium ions as well as one more also, negatively charged proteins. Negatively charged proteins. So this is known as polarized state here. So what is the meaning of polarized state? Means whenever the plasma membrane is in resting state, that is without any stimulus, that membrane will be permeable to potassium but nearly impermeable to sodium ions as well as negatively charged proteins here. So because of this thing, the neuron will have charges. The neuron will have charges. What are the charges present outside means here? Outside charges are positive. Positive charges. Same thing. This is axon and this is axolemma. Okay. This is extracellular fluid. It means outside the neuron. Extracellular fluid will have a charge of positive ions, and inside it will have negative charges. Negative charges here. So this is known as intracellular fluid, or even it is also known as axoplasm. Axoplasm here, because this is axon, so here inner side it has a fluid called axoplasm here. So whenever the membrane will be in this way, this is known as polarized state. Polarized state, or even it is also known as resting potential, or even it is also known as resting potential here. Next. So what is the reason? What is the reason of having positive charges outside and negative charges inside when the membrane is in the polarized state means here? The reason is outside there is high levels of sodium ions, very high levels of sodium ions, and low levels of potassium ions. Okay, so what is the reason of having positive charges outside means here? The outside, that means in the extracellular fluid, it has more concentration of sodium ions and less concentration of potassium also present. What about inside, that is axoplasm? Axoplasm will have high amounts of potassium. Not only potassium, even it has high amounts of negatively charged proteins. Negatively charged proteins as well as low levels of sodium ions. It means potassium and sodium is present on both sides, but they are differing in their concentration here. Outside more sodium ions, which imparts this positive charges here, but inside there will be more amount of potassium as well as negatively charged proteins. This negatively charged proteins imparts this negative charge here. So this is known as polarized state here. What is the meaning of polarized state? Means having positive charges outside and negative charges inside. This will happens only during resting stage. What is the reason of having this type of different charges means here? The reason depends on permeability. Permeability means during resting stage, the membrane is permeable to only potassium but nearly impermeable to sodium as well as negatively charged ions. As a result, it leads to more amount of sodium outside and less amount of sodium inside. But potassium will be more inside, outside it will be less, as well as proteins also. This is known as polarized state. Next, here some definition given in the textbook. Resting potential difference. What is that resting potential difference means here? Important definition. The electrical potential difference. The electrical potential difference across resting plasma membrane. Across the resting plasma membrane is known as resting potential here. It is known as resting potential. Next. So because of this different concentration, it causes or it develops concentration gradient, because there is a difference in concentration on two sides. For that reason, it develops. It develops a concentration gradient. Okay, so whenever there is a difference in concentration gradient, that time only the neuron will be present in the form of polarized state. So this maintaining of polarized state makes the neuron excitable. Next, who is maintaining this concentration gradient means here? First of all, in neuron membrane, has, this is NCERT line, it has different ion channels. Neuron membrane has different ion channels. This is statement question, different ion channels, and these different ion channels, they are selectively permeable. They are selectively permeable to same ions, means no, it is actually different ions. This point is important. So neuron membrane has different ion channels, and these different ion channels are selectively permeable. They are selectively permeable to different ions. Okay, what are those ion channels? For example, there are different types of ion channels are there. The ion channels may be, number one, it may be passive channels. It may be passive channels. Passive channels means they will allow the ions from high concentration to low concentration without the cost of ATP. That is passive channels. So some channels will be passive, and some channels are known as voltage gated channels. They are known as voltage gated channels. These voltage gated channels will open whenever some stimulus will be given to the neuron. And the third one is here, active channel. Third one is here, active channel. That active channel is known as sodium potassium pump. Active channel is known as sodium potassium pump. So this pump is actually active. Active meaning is here, it pumps the ions at the cost of ATP. Here. So like that, there will be different types of ion channels which are present on the neural membrane. But these ion channels are selectively permeable to different ions. Selectively permeable to different ions means, for example, there are two types of voltage gated channels are there. What are those two types of voltage gated channels means here? Sodium voltage gated channel, potassium voltage gated channel. Sodium voltage gated channels allows only sodium, and potassium voltage gated channel allows only potassium. That is known as selectively permeable to different ions here. Out of these three channels, which channel is responsible for maintenance of this one? Maintenance of this resting potential or concentration gradient means here the concentration gradient, across the membrane this point is important point. The concentration gradient across the membrane is maintained. Whenever there is a maintenance, that time only it will exist in the form of polarized state here. If there is no maintenance means here, the membrane cannot maintain the polarized state. Whenever it is not maintaining the polarized state, that time it may become non-excitable state here. So the membrane is maintained by a pump called, what do you call that pump means? That previously discussed, that is known as active pump. It is maintained by active pump. This active pump is also known as sodium potassium pump. So which of the following transport or ion channel maintains the membrane potential or maintains the concentration gradient? Means the answer is sodium potassium pump or active pump here. What is the role of this pump? What is the role of this pump means? For example, this is membrane. Over the membrane, this channel will be there. Okay, this is sodium potassium pump. So this sodium potassium pump, it allows 3 sodium ions outside, 3 sodium ions outside, and simultaneously it allows 2 potassium ions inside here. So whenever this channel plays an important role in movement of 3 sodium ions outside and simultaneously 2 potassiums inside at the cost of ATP. ATP is important. At the cost of ATP, that is known as sodium potassium pump. Because of this movement here, the pump can maintain these charges. Because of this, always the outside will have positive charge and inside will have negative charge. So this is known as maintenance. Then the membrane is said to be polarized. This is a repeated question. This is a repeated question here. Then what is the role of this sodium potassium pump? This pump will pumps 3 sodium ions from where? From exoplasm to outside. It means from inside to outside. From exoplasm to outside or extracellular fluid also correct. Whereas simultaneously it pumps 2 potassium ions from, in the reverse manner, from outside to exoplasm or inside at the cost of 1 ATP. 1 ATP here. So this pump is very, very important for maintaining of polarized state. Yes. So just you observe this picture. This is membrane. This is the membrane and this is inside the cell. Inside the cell means here this is exoplasm. This is exoplasm, whereas this is outside the cell called extracellular fluid. So what is the charge of extracellular fluid? It is positive. It is positive charge and inside charge is negative. What is the reason of positive charge towards outside? The reason of positive charge outside is because of high concentration of sodium ions. High concentration of sodium ions. Whereas inside it has negative charge. The negative charge is mainly due to potassium ions also there, as well as some negatively charged proteins also there. This one. This is the protein here. So due to increased levels of negatively charged proteins. Here. So outside sodium will be more and inside potassium will be more, as well as negatively charged proteins also more. But this negatively charged proteins, it imparts negative charge towards the inside and sodium ions imparts positive charge towards the outside. Automatically the membrane is said to be polarized. It is said to be polarized state here. So whenever it is existing in the form of a polarized state, that time it is ready to take the stimulus here. So I said now this membrane is having, this neural membrane will have different types of ion channels. Those different types of ion channels, they are selectively permeable to different ions here. So this is a type of ion channel. This ion channel is specific to sodium. So you can take this channel as sodium ion channel. It is actually a sodium ion channel. This sodium ion channel, it is allowing the positive charge, that is sodium ions, from outside to inside. From outside to inside here. Next, this is one more channel that is just like a passive channel. That previous one is passive channel, and this is voltage gated channel. This voltage gated channel will get opened whenever you are giving some stimulus. So in response to the stimulus, this voltage gated channel will get opened, and because of that opening here, the ions may diffuse. Suppose this is a sodium voltage gated channels, means the sodium ions will diffuse from outside to inside. Suppose if this ion channel is a potassium voltage gated channel, that time the potassium ions will diffuse from inside to outside here. So this is the second type of channel called, this is voltage gated channel here. And next one, this is known as sodium potassium pump, third type. Sodium potassium pump. Even this sodium potassium pump is also present on the same plasma membrane only here, but the role of this one is, already we have discussed here, it allows 3 sodium ions out and 2 potassium ions in at the cost of 1 ATP. This is a very, very important point. So by seeing this animation easily you can understand here. In this one, there is a movement of 3 sodium ions outside, and simultaneously 2 potassium ions, they are moving towards inside at the cost of ATP. That's what it is known as active channel. What's the role of this active channel? This active channel involves in maintenance of that concentration gradient, and it always maintains the membrane in resting state or in polarized state here. Now we are going to give some stimulus. So till now we have discussed about the membrane when it is in the absence of stimuli or in the resting stage. Now here we are going to give stimulus here. So this is a polarized membrane. This is a polarized membrane here. So polarized state means always it contains positive charges outside and negative charges inside. Okay. Now we are considering this one as A site and this one as B site. And this is B site here. Then some stimulus is going to give. The stimulus is going to give only at a particular point called A. Because of this stimulus, there will be opening off. So whenever the stimulus will be given, it leads to opening of sodium voltage gated channels. It leads to opening of sodium voltage gated channels. So whenever these channels gets open, automatically it increases the permeability. It increases the permeability of the sodium. Here, this is important. It increases. Earlier, the sodium was impermeable, but now because of stimulus, here the permeability has increased here. So first thing is, this is polarized state. In this polarized state, some stimulus is given at a particular point called A. Because of this stimulus, it causes a opening of channels. Here some channels will be there. These channels gets opened. The channel name is sodium voltage gated channel. Because of opening here, the outside sodium will enters inside. Here, it means the permeability has increased. So it increases the permeability of sodium, and it leads to rapid influx or outflux. As the sodium is moving inside, it means influx. It leads to rapid influx of sodium. Important point here. So why do there is a rapid influx of sodium? Means rapid influx of sodium will causes due to stimulus. Stimulus induced opening here. Next, rapid influx of sodium leads to reversal of charges. Here. So this is A site and this is B site here. So earlier it was positive outside, but now it has become negative. Why do it has become negative on outside means here? Because of rapid influx of sodium. Wherever this type of reverse will takes place, that is known as depolarized. So now the A side gets depolarized, and what about the B side? The B side is normal only, still it is in the polarized state only. The B is in the polarized state only. So whenever there is a reversal of charges, because of reversal of charges, it generates action potential. So why do the action potential has generated means action potential has generated due to reversal of charges here. Okay, what is the definition of action potential? The electrical potential difference across the plasma membrane at site A. Where do the difference has developed? It has developed only at site A. So that is known as action potential. Here. So whenever there is an electrical potential difference occurs across the plasma membrane, not throughout the plasma membrane, only at one particular site called site A, it is known as action potential. And that action potential is also known as nerve impulse. It is also known as nerve impulse. Now, where do this nerve impulse or action potential has generated? It has generated at A site. It has generated at A site here. Now the next step is actually, what is the topic here? The topic is generation of nerve impulse as well as conduction of nerve impulse. Now the action potential has generated, and the next step is here, this action potential has to transmit or it has to conduct to the next site. Next site means to adjacent site called B site here then. So because of generation of this action potential, have you seen there is a difference? Here negative charges are there, here positive charges are there. Here also positive charge, negative charge. Whenever this type of difference will be there, it generates electric flow here. It generates electric flow. Then next step is, this is conduction. So how the conduction will takes place means here, the electricity will flows from, this is B side and this is A side. Electricity is passing from always positive to negative. Here also like this, positive to negative. It means towards the outside, the flow has done from which side to which side? This is important point. From which side to which side here? Yes, Niharika. New one only, Niharika. Better to follow new NCRT, it will be good. So don't follow old one here, better to follow the new one. Here how the electricity is passing? The electricity is passing from B to A site. It is passing from B to A site outside, and whereas inner side is A to B site. Here, this point is important here. So this step is here conduction. In this conduction here, the action potential, because of action potential at A here, it creates a different charges. These different charges will generate electric flow, and the electric flow is passing from B to A outside and A to B towards inside here. Then after that, Now the charges are reversed or they are returned like this. Now the A site is said to be repolarized. Now the A site is said to be repolarized. Why do we are calling the A site as a repolarized means? Because they got their charges back here. So just like a polarized state, here also they got positive charges outside and negative charges towards inside. Then what about the B site? B site has become reverse. This is B site. So in this case, the B site is, now where is the action potential? Now the action potential reached to, earlier it was at A site, now the action potential reached to B site. Okay, it is reached to B site here. Then the B automatically become depolarized here. Okay, this is known as conduction. In the same way, the mechanism will continue throughout the neuron membrane. The same thing will happen. So just you observe here, earlier it was positive charges outside because it is present in the polarized state. Because of stimulus here, the positive charges has become negative, and later on the action potential is passing. The action potential is passing from one side to another side. For example, imagine this is A side, next B side, next C, D, E, F, and further here. So like that the action potential is passing from one side to another side, and the previous sides automatically become polarized, and the previous sides will become ready to take one more time the next stimulus. So like this is also same. What are these dots here? These are channels. These are the channels. Whenever the action potential, that red color mark is action potential here. Because of this action potential here, the ion channels are working, and so that there is a reversal of charges. Reversal of charges will take place. This process is known as conduction of nerve impulse. This is known as conduction of nerve impulse. Okay, this is NCERT picture here. In this NCERT picture also they are showing two lines. What are these two lines here? This one, this is channel. This is an ion channel here. So this red mark is action potential here. So due to action potential generation, outside is having negative charges and inside is having positive charges. Next, after that there will be developing of electric circuit. How the circuit is passing here? There is a movement of electricity from B to A outside, and inside will be A to B. As a result, the action potential is transmitting to next site called B site. Next site to B site here. So it is a representation of impulse conduction through the axon. Here. Next, here in NCERT one line given. This is actually important one. The rise in stimulus induced permeability is extremely short-lived. This is important statement question here. The rise in stimulus induced permeability is extremely short-lived. It means here permeability of sodium. permeability of sodium is extremely short-lived. Meaning is, whenever the membrane wants to be in the polarized state, that time it should be impermeable to sodium. But here during stimulus time only, the membrane will become little bit more permeable to the sodium. But that permeability will be short-lived. It is not for long time, it will be short-lived here. So that is the meaning. The rise in stimulus induced permeability of sodium is extremely short-lived, and after that there is increase in permeability of permeability of potassium. So first sodium, but that sodium permeability will be short-lived, and later on there is increase in permeability of potassium and causes repolarization or depolarization. Here it causes repolarization, and this repolarization is due to influx or outflux. It is mainly due to efflux of potassium. So this point is very, very important here. Why do the depolarization is taking place? Depolarization is taking place due to influx of sodium. Influx of sodium causes depolarization. Now here outflux or efflux of potassium. Potassium causes repolarization. These two are important points. It means in this picture, the A is in which state? It is in depolarized state. The A is in depolarized state here. So why do it is in depolarized state? Means earlier there was an influx of sodium, but now here there is an efflux. It means here one channel will be there. This channel is known as potassium channel. This is actually potassium channel. This potassium channel will opens after some period, so that it results in movement of potassium out. Because of potassium out, automatically the A site will become normal. The A site will become normal because of efflux of potassium ions from inside to outside here. Okay, inside to outside. So the A site has become repolarized here. So this is the cycle. So how the cycle has started? First polarization, next depolarization, and then repolarization. This cycle same continues along the length of neuron, here. So once again, important points. Why do depolarization occurs? Depolarization occurs due to stimulus. And stimulus causes increased sodium permeability. Increased sodium permeability. So increased sodium permeability means here sodium influx. It causes sodium influx. Match the following. Sodium influx causes depolarization. Okay, sodium influx which causes depolarization. Same like that. Why do the repolarization occurs? Why do the repolarization occurs? Means repolarization occurs due to due to closure of sodium channels, due to closure of sodium channels. In the last slide only I said here, the rise in stimulus induced sodium permeability is extremely short lived. It means these ion channels will open only for short duration. So immediately it gets closed here. So due to closure of sodium channels and simultaneously opening of potassium channels. Potassium channels causes increased permeability. This time there is an increased permeability too. It causes increased permeability to potassium. It means potassium efflux will take place or outflux will take place. Outflux. So potassium outflux causes repolarization. Sodium influx causes depolarization, but potassium outflux causes repolarization. So like that the entire membrane will undergoes depolarization, repolarization like that, and it will continue. The same action potential now it will reach us to that action potential. This is axon. What is axon? The axon is a long fiber which is arised from the cell body, and distal end of this axon, it is branched. Distal end is branched, and this branched structure we are calling as telodendria. And to this branches it has a swollen knobs. These swollen knobs are known as synaptic knobs. It is known as synaptic knobs, and this synaptic knobs has some vesicles, and these vesicles are filled with neurotransmitters. Filled with neurotransmitter. Now where is the action potential? The action potential has generated starting, and it is going on continue here, and now it has reached to end. So the procedure will be same from starting to till end, but whenever the action potential will reach us to the end, here this is different. Now that topic we'll discuss tomorrow here, that is transmission of impulse. Transmission of impulse through the synapse. So that topic we'll discuss tomorrow. Thank you very much.
Good morning everyone. So yesterday we did the technique how we can draw structures, right? I told you 3 main points to draw any structure in the inorganic chemistry or organic chemistry also. I told you first of all you should know how to find valence electrons. Then second point is you should know which element can make maximum 4 bonds or which element can make more than 4 bonds as well. Like I told you, second period elements, beryllium, boron, carbon, nitrogen, oxygen, these elements as central element cannot make more than 4 bonds because of absence of d orbital, right? And silicon, phosphorus, sulfur, chlorine, bromine, iodine, xenon, these elements can make more than 4 bonds because of presence of d orbital, right? So with these 3-4 points you can make easily the structures. And one more point, most of the time surrounding atom will be hydrogen or oxygen or halogens. So hydrogen as a surrounding element will always make 1 bond, right? We know that, very easy thing. Halogens also will make 1 bond only, and oxygen will make how many bonds? 2 bonds. So with these 2-3 points easily you can draw all of your structures, right? Clear? So let us see 1 or 2 more examples, then we will move ahead with the topic hybridization. Fine. For example, I am taking here some more examples. Imagine I am having here phosphate ion PO4 3 negative, right? I want to draw its structure, phosphate ion. The first thing is you have to identify that phosphorus is my central element and write the valence electrons in the phosphorus. 15th group element, so it have 5 valence electrons, right? Now write the phosphorus, and now you can write the surrounding elements. Like I am writing 4 oxygen are present, so I am writing oxygen like this. It's fine, you can write anywhere. We are not here very particular about geometry or structure, just we are finding how many bonds it is making and how many lone pair will left on central atom. So as they are giving PO4 3 negative, so it is very common. You can check oxygen is more electronegative, so the negative charge will be present at oxygen. And now O negative is having 7 valence electrons, so it will make one bond. O neutral O will make 2 bonds, but O negative will make only single bond, right? So you will see O negative is making here single bond and single bond, right? Neutral O will make what here? Double bond. That's it. P was having 5 valence electrons. Out of 5, you can see all the 5 are used in making these 5 bonds. So no valence electron left on the phosphorus. That's why there is no lone pair. That's it. It is finished up to here. Clear, right? Similarly, we can take one more example. Next one, if I say I am having here, you can imagine we are having here NO2 negative nitrite ion. We are having here nitrite ion. So again, same way we have to move. Nitrogen here is having 5 valence electrons belonging to 15th group. It is having 5 valence electrons, right? So write the nitrogen first, right? And now I am writing here oxygen and oxygen. Fine. You see NO2 negative. So definitely one of the oxygen is what? Negative charged. So the negative charge oxygen will make what here? Single bond, and neutral oxygen will make here double bond. Fine. Now out of the 5 valence electrons, nitrogen used its 3 valence electrons in making bond. So how many left? 2 left. Okay. The 2 valence electron left will be here as a lone pair. Very easy. Getting my point? Sure, right? Okay, great. Now the next thing, if I say here, if I take here one more example, imagine I am taking here SO3 2 negative sulfite ion. Same way, if I take here sulfite ion, so here sulfur is having how many valence electrons? Sulfur is having here 6 valence electrons belonging to 16th group. So I will write sulfur here and I will show oxygen. 3 oxygens are present. So I am showing here 3 oxygens and 2 negative, means 2 of the oxygens are here negative charged. Right? Clear? Now we know that the O negative will make which bond? Single bond. Single here, single here. Neutral oxygen will make which bond here? Double bond. Neutral oxygen making here double bond. Right? Clear? And out of the 6 valence electrons, tell me, sulfur used how many valence electrons in making bonds? So sulfur used its 4 valence electrons in making bond. So how many left? 2 left. So those 2 you see will become lone pair. Getting my point? Sure. Count the valence electron on center atom. Write the surrounding atoms. Make bonds accordingly. Central atom used how many valence electrons in making those bonds? Remaining electron will become lone pair. Getting my point? Right. Right, Rohit? One lone pair. Very good. Fine. Now, one thing you have to be very careful. Like if I take this time, I am taking nitrate ion, NO3 negative. NO3 negative. Here we have to learn one thing. Okay? Look at this. In nitrate ion, nitrogen here, if you look carefully, nitrogen here is having 5 valence electrons because it is 15th group element, right? So I will take here nitrogen, right? I am having what here? Oxygen, oxygen, oxygen. 3 oxygens and NO3 negative. So one of the oxygen is having a negative charge. Simple thing. 5 valence electrons in nitrogen, and I have put the surrounding atoms as well. Now I know the O negative will make single bond. That's very clear. Neutral oxygen will make what here? Double bond. Right? And neutral oxygen will make what here? Double bond. Right? I want to ask you, is it right or I am doing some mistake here? This structure written is right or we are doing some mistake? Whenever your central element is making more than 4 bonds, you have to check the point number 2 and point number 3. In point number 2, I told you which elements cannot make more than 4 bonds. And in third element, I told you which elements can make even more than 4 bonds. Right? So in this case, if you check carefully, you see nitrogen is making here 5 bonds. Right? I told you very clearly second period elements cannot make more than 4 bonds. That means we are doing some mistake. Nitrogen is making 5 bonds. It is wrong. It is not possible. Okay. So we will modify a little bit. Imagine up to here. Up to here, if you imagine, you see nitrogen made 3 bonds. And after making 3 bonds, 2 electrons are remaining. So I am showing those 2 electrons here only. Right? As nitrogen cannot make here double bond, and here oxygen is having only 6 electrons. So to complete the octet of oxygen, it will make a coordinate bond here. It will not make a double bond. It can't make double bond here. It will make a coordinate bond. That's why nitrogen will put both of these electrons here and will make a coordinate bond. So you have to be careful about it. Nitrogen cannot make more than 4 bonds. Whenever this situation comes, remove that double bond and make a coordinate bond here. Right? So one more thing we can learn from here, that whenever, we can write one more point, very important point. If nitrogen is making 4 bonds, if nitrogen is making 4 bonds, the 4th bond will be a coordinate bond. The 4th bond will be a dative bond. You can say will be a dative bond or coordinate bond. Both are same thing. Right? This will help you to solve one more type of questions. Sometimes they ask which of the following is having a dative bond or coordinate bond. So if you see a structure in which nitrogen is making 4 bonds, definitely it is a coordinate bond. Getting my point? If I gave you one more example for this, imagine I am having here N2O5. I am having N2O5. I am giving the structure, don't worry. It's N here, it's O here, it's N here, it's O here, O, it's O. Can you tell me? Let's see if I want to ask you, in this structure, coordinate bond is present or not? In this structure, coordinate bond is present or not? Yes, coordinate bond is present here. If nitrogen is making 4 bonds, coordinate bond is present here. Right? Clear? So if nitrogen making more than 3 bonds, the 4th bond is a coordinate bond. Right? So you can identify. Clear, bacha? Right? In NO2 plus, do not think it is making coordinate bond. In NO2 plus, N is plus. So those 4 bonds are there. Those are normal bonds. When atom nitrogen is neutral and making 4 bonds, so definitely one is coordinate bond. Clear? Good. That's it. Now we have done a lot of practice about structures, right? So now I'm starting the next topic, hybridization, right? We will see first the shortcut, how to find hybridization, and then we will see the details also, right? So first of all, shortcut is very important because you have to save time in your NEET exam, and then we will see the details also, right? It's a very common topic and very important topic from NEET point of view. In every chapter, there are so many structures and they're asking the hybridization of this atom, this atom, this atom. So how to find the hybridization, right? So I am starting the shortcut only because it is the most important question of this topic. How to find hybridization? You have to do one thing. You have to count number of sigma bonds formed by that atom for which you are finding hybridization, and plus you have to count number of lone pairs. This sum on that atom will tell you its hybridization in very short time, right? Imagine this sum is coming 2. So that means that element is sp hybridized. If this sum is coming 3, you will say that element is sp2 hybridized. If this sum is coming 4, you will see that element is sp3 hybridized. If it is 5, that will be sp3d hybridized, and if it is 6, it is sp3d2 hybridized. If it is 7, then it is sp3d3 hybridized. So just by counting the number of sigma bonds plus lone pair formed by that atom, you can tell its hybridization in any structure, right? First of all, these are the main part. When you will go in chapter coordination compounds, you will see some extra part, like if there are 4 things, so instead of just sp3, dsp2 also possible. If you have 5 things, then dsp3 also possible. If there is sp3d2, then it is d2sp3 also possible, right? And if it is sp3d3, then sorry, it is a fine. Okay, so mainly these things we had to remember. Clear? So normally up to now it is enough. Okay, this type of things you will see sorry, this type of things you will see in coordination compounds. In your chemical bonding chapter, this is enough to remember, right? So we will see how we can answer our questions very easily using this technique. I'm saying just count the number of sigma bonds and number of lone pairs on that atom for which you want to find the hybridization. And in most of the cases, they will ask hybridization for the center atom because it will be hybridized definitely, except very few cases which Drago rules tell us that in which case the center atom is unhybridized, but it is not relevant because the NEET does not ask that type of questions. Fine. So we will take example, like if I am saying example number 1, I am taking the BeCl2. BeCl2 is having this structure. You can easily draw the structure using the technique which we discussed. Okay. If I take example of BeCl3, so here it is BeCl3, right? Now third one, if I take example of CH4, here we have CH4, right? Next one, if I take example, you can say ammonia. So we are having ammonia like this. Now we can have here the fifth one, water example. It's water, fine. And then we can have PCl5 also. Fine. So first of all, we will try to find the hybridization for these elements. Clear? If you know the structure, we did the last topic. So using the structures, you can find hybridization very easily. There are some other methods as well. We will discuss about it. But the best method is using structures, right? So first of all, if we try to find the hybridization of beryllium, we count how many sigma bonds are present. 2 sigma bonds are present. There is no lone pair on beryllium. So sum is coming how much? 2. Sum 2 means it is sp hybridized. It is sp hybridized. Fine. If you go further, in this case beryllium is having 3 sigma bonds plus 0 lone pair. Sum is coming what? 3. 3 means we know it is sp2 hybridized. So you see, it will take just seconds to answer. Now in CH4, if we go, in here carbon is having 4 sigma bonds plus there is no lone pair, so sum is coming 4. 4 means what? 4 means it is sp3 hybridized. Right? Very simple. Now in this case, nitrogen is having 3 sigma bonds and plus is having 1 lone pair. So 4 is coming. 4 means what is that? 4 means again is what? sp3 hybridized. Nitrogen there and carbon here both are sp3 hybridized. But here it is having all 4 bonds, no lone pair. But in that case, 3 sigma bonds and 1 lone pair. Okay? Sometimes they ask questions like 2 atoms can have same hybridization only when they have same number of sigma bonds and lone pair. No. Because here you can see in these carbon is having 4 sigma bonds with 0 lone pair. It is sp3 and this also sp3 nitrogen, but it is having 3 sigma bond and 1 lone pair. Their shape is different, but the hybridization is same. We will discuss this part later. Oxygen we can see now. Here oxygen, 2 sigma bonds plus 2 lone pair. Total part is what? 4. 4 means what hybridized again? sp3. Clear? Easy. Now this one P, phosphorus here. 5 sigma bonds, 0 lone pair. So 5 total sum is coming. And 5 means what? It is sp3d hybridized. It is sp3d hybridized. Is it clear? Right? Easy. Fine. Now the next part we can see. We can go to the next example. Next example is, you can take here SF4. Right? We can take SF4. If you go for the structure, sulfur is having how many valence electrons? 6 valence electrons, right? 6 valence electrons are present. I am writing fluorine as surrounding atom. You know fluorine makes single bond as surrounding atom. So single, single, single here, single here. Out of 6, 4 are used in making bonds. How many remaining? 2 are remaining for sulfur. So it becomes a lone pair. Clear? So if you count for sulfur, okay, this is it. We will see the hybridization later. First I am writing. Next, if you can go, you can take ClF3. Right? ClF3 here, Cl present, F3. So I am putting here 3 fluorines. Cl having 7 valence electrons. It is using its 3 valence electrons to make bond, and remaining 4 become what? 2 lone pairs. Fine. The next case you can take here is XCF2. Xenon have 8 valence electrons. Out of 8 valence electrons, 2 are used in making bond. Remaining 6 become what? Lone pair. Right? And one more I will take. Here you can see it is I F7 I can take. Right? Okay, before F7 we can take SF6 also. We are having SF6. Right? In SF6 sulfur have 6 valence electrons. So I will keep first of all the surrounding atoms here. Now every fluorine will make what bond? Single bond. So there is nothing left. All 6 valence electrons of sulfur are used. So there is nothing, no lone pair present on sulfur. So we are practicing how to make structures as well as the hybridization part. Clear? The next thing you will see, I can take here BrF5, right? So we are having bromine with 7 valence electrons and the surrounding atom are fluorine. Okay. So after making the 5 bonds with fluorine, 1 lone pair will be present on bromine, because bromine have 7 valence electrons. After making the 5 bonds, 5 are to used, 2 are remaining, so 2 electrons become the lone pair one. Fine. And the next one is XCF4. Xenon have 8 valence electrons and we are keeping here 4 valence electrons. So, sorry, 4 fluorines. Out of 8 valence electrons of xenon, how many are used here? How many used here? 4 used. So 4 valence electrons remaining become 2 lone pair. Fine. So I will show it as lone pair. Clear? So In that way, we made the structures. There are other techniques as well to find hybridization, but the best one is using structure. Because if you know how to draw structure, it will help you in many ways, not only in hybridization, many ways, right? So now you find sulfur here is having 4 bonds, 4 sigma bonds plus 1 lone pair, total 5. 5 means you know it is what? sp3d. In ClF3, if you see, Cl is making 3 bonds plus 2 lone pair, again 5. 5 means what? sp3d. Right? In this case, if you see xenon is having here 2 sigma bonds and 3 lone pair, again it is 5. It is sp3d. Okay? So total sum is coming 5. So all of these, this one, all are sp3d hybridized. Even PCl5 was sp3d hybridized. Right? Clear? The next part is, you can see sulfur here. In this case, sulfur is having 6 sigma bonds plus 0 lone pair. Total sum is coming how much? 6, right? So you can see here, the hybridization is sp3d2. If sum is coming 6, it is sp3d2. It can be d2sp3, but not in this case. In such cases, in coordination compound, you will find that part, right? Next part you will see bromine here. 5 sigma bonds plus 1 lone pair. Again 6 coming. 6 means what here? sp3d2. Fine. In this case, xenon, if you count here, this xenon is having 4 sigma bonds plus 2 lone pair. How much coming? 6. sp3d2. Clear? This way we can easily find the hybridization. One last example I will say here. Okay, I hope it's clear up to now. The next thing is IF7. The next example is IF7. Right? Here you will see IF7. I have how many valence electrons? 7 valence electrons. So first of all, put all the fluorines surrounding the iodine. Okay? And then here we are having, so I am putting 7 fluorines. Someone is asking doubt, what is difference in D2sp3 and sp3d2? Fine. Okay, so I will tell you, it is not required for now, but yes, you can listen to me carefully. What happened? Imagine you are using your 3D, look at this. You are using your 3D orbitals, then you are using 4s orbitals, and then you are using 4p. So when you are using your 2 of the d orbital from here and s orbital from here and p orbital from here, so it is d2sp3, means you are using d orbitals from the inner shells. Like I am using d from my 3rd shell, I am using s from 4th shell and p from 4th shell. So d is getting used first, then s, then p. Getting my point? So d2sp3. Whereas imagine in some of the cases you will see 4s I am using 4p and I am using 4d. So I am using my 4s s orbital. From here I am using my 3p orbital, and from here I am using my 2 of the d orbitals. Right? So I am using the d orbitals which are coming later. I am using here the backside d orbitals. So I will say sp3d2. First s, then p, then d is coming, and I am mixing them to hybridize. So in this case you will say this is sp3d2 hybridized and this is your d2sp3. Getting my point? If you are using d orbital from the inner shell, it is d2sp3. If you are using your d orbital from the outer shell, it is sp3d2. I think you are clear, beta? Yes? Right. Now, yes, good thing. You are asking one more question. When we intermix the orbitals, I will discuss these things first of all. Still, you can listen one more line. When you mix the orbitals, their energy should be approximately equal or similar. It is not exactly same. It should be similar or very less energy gap. Okay? It is not like you cannot say energy should be same only. When I mix, there is definitely if I mix 4s and 4p. So 4s and 4p also have difference in energy. 4s, 4p, 4d I am mixing, so there is little difference, but that energy difference is not so much. So you can mix them. When energy gap become high, then it is not possible to mix. But with little energy gap, we can mix. Okay? Yes, bacha? You can mix it, no doubt. When there is little energy gap, like I am saying, I am using 3D, I am using 4s, 4p. So this energy gap here is not so high. You can mix these. Even if you say I am using 4s, 4p and 4d, then also energy gap is low only. So you can mix these. When energy gap become very high, then you cannot mix it. Okay, bacha? Getting my point? You are saying very right line that energy should be almost similar or little energy gap. But that little energy gap is present in there, so you can mix. You can mix 3D, 4s, 4p, or you can mix 4s, 4p, 4d. It depends on the atom which you are using in hybridization part. Getting my point? Clear? Okay, sure. Now you see here, we are taking the IF7 like. So here iodine is present, having 7 valence electrons. You will see F2 will make what? Single bonds with every fluorine, right? So here iodine, you can see beta, iodine is making 7 sigma bonds, and you can see none of the lone pair is present because all the 7 valence electrons are used in making the bond. So nothing left, so no lone pair is left. So here we have, sum is how much? Sum is 7. Sum is 7, so I can say total sp3d3. How many orbitals overall? Overall 7 orbitals. Total how many orbitals? 7. So sp3d3. Clear? That's it. This is how you can find the hybridization for your compounds. Clear? Done everyone? Now I will share one more shortcut. Okay? And tomorrow we will discuss the details. So I'm sharing one more shortcut to find the hybridization, because most of the time they ask this type of question, find the hybridization of central atom. So one more shortcut I am writing here. Okay, how we can find hybridization without even making structure. Every shortcut has some limitations, but the structure method is not having any limitation. Fine. So here I am saying the formula is 1 by 2 here. Number of valence electrons on central atom, right? Half is there, number of valence electrons of central atom, or you can say for which you are finding hybridization. Okay, number of valence electrons of atom, mainly we find for central atom, so I am writing for central atom, right? I am finding its hybridization, plus number of monovalent atoms attached to that atom. Number of monovalent atom attached. Okay, or directly I can say number of monovalent atom present. Okay, directly. Why to write so big? So you can say number of valence electrons. I am removing this part also simply because they will ask for central atom only. Even if they ask for some other, in big structures, many atoms can be hybridized, no issue. Okay. So in this case, I am saying the atom for which you are finding hybridization, you have to see number of valence electron for that, and then for number of monovalent atom. Monovalent atom means which will make a single bond with our central atom, sorry, right? This you can apply. I can make this formula a little big. If there is a negative charge on our ion, so you will add electron. If there is positive charge on ion, you will remove electron. Okay, so I will show you some examples. Clear, bacha? So for example, like I took already, I took the BeCl2 example. So without structure, how we can find that? So first of all, tell me the beryllium is having how many valence electrons? Beryllium is having 2 valence electrons. And you can see here the chlorine is a monovalent atom. Mainly you will see halogens will come or hydrogen. So these things, hydrogen and halogens, are monovalent. Whereas oxygen you have seen make 2 bonds, double bond. It is a bivalent atom. Clear? So here hybridization is what? Half 2 valence electrons plus 2 is the number of chlorine, because chlorine are monovalent atom. So 4 divided by 2, how much sum is coming? 2 is coming. 2 means what hybridization? sp, hybridized. Clear? So without using the structure, we are finding hybridization in such kind of cases, right? Next example I can take here, like if I am saying BCl3, right? In BCl3 I want to find hybridization of boron definitely. So it is 1 by 2. How many valence electrons? 3. There are 3 valence electrons, right? And next you will see there are 3 monovalent atoms. Chlorines are monovalent atoms which are connected to the boron. So 3 plus 3 is 6 divided by 2, how much coming? 3. 3 means what is the hybridization? sp2. 3 means it is sp2 hybridized. I hope we are clear, right? Next example I am taking here, one more. Imagine I am having SO3. By this method, try for SO3 beta, and you can go for S2 also. In S2 find for oxygen, right? Definitely here you are finding for sulfur, here you are finding for oxygen, center atom. Okay, fine bacha, fast. Find the hybridization for sulfur in SO3 and find the hybridization of oxygen in water. Yes, answer me fast. Look at this one. If I will take hybridization, it's 1 by 2. Sulfur have how many valence electrons? 6. Oxygen is a bivalent atom, it is not a monovalent atom, right? So I will say 6 plus 0. So 6 divided by 2, how much coming? 3. 3 means what? sp2. I'm saying once again, why I am taking 0 here? Because oxygen is a bivalent atom, not monovalent. I have to count monovalent atom only. Clear? Very good. Right answer. Now H2. Here the hybridization is what? 1 by 2. Oxygen have how much? Oxygen have how many valence electrons? 6. And it is connected to hydrogen. Hydrogen is monovalent atom. It's fine, right? So I will add it. 6 plus 2, because monovalent atom how many? 2. So 8 divided by 2, 4 is coming. 4 means what? It is sp3 hybridized. Are we clear about this thing? Yes, I hope we are clear. Okay, we will see more examples like this tomorrow by this formula. I will take examples of cations and anions as well. Okay, so this is it for today. Because of some work, I am leaving you 5 minutes early. Fine. So remaining part we will see tomorrow. Okay, bacha. Fine. I hope these things are benefiting you. So just work hard and revise all these points. Okay, bacha. Bye. See you.